Hello, and welcome to the Cognitive Revolution, where we interview visionary researchers, entrepreneurs, and builders working on the frontier of artificial intelligence. Each week, we'll explore their revolutionary ideas, and together we'll build a picture of how AI technology will transform work, life, and society in the coming years. I'm Nathan Lebenz, joined by my co-host, Eric Torenberg.
Hello, and welcome back to the Cognitive Revolution. Today, I'm speaking with Amelie Schreiber, a computational biochemist and AI researcher working at the forefront of applying the latest AI models and research techniques to some of the most complex and impactful domains imaginable: drug design, protein network engineering, and broadly unlocking the secrets of biological systems. In all honesty, this episode has given me more future shock than perhaps any other episode we've done. While traditional analytical methods are not well suited to the crazy complexity of biological systems, and as a result, there's still much more that we don't understand about biology than that we do, as Amelie makes clear, modern AI architectures are perfectly suited to take advantage of biology's massive datasets. And thus, a new wave of AI models are poised to dramatically accelerate the process of scientific discovery in biology. Starting with how we understand protein and other cellular structures and the physical interactions between them, and likely soon zooming out to help us understand how our cells, tissues, and bodies function at higher levels. As you might expect, given the incredible complexity of biology, if you're not already well versed in the subject, the first hour will feature a number of new technical concepts. And while Amelie and I do our best to explain them all clearly, this episode, for me at least, did require multiple rewinds for full comprehension. I think it might help to keep in mind the distinction between static structural analysis and dynamic conformational analysis, which allows for molecules to change shape as they interact with one another as you listen. Remember, too, that all the models we discussed today are really rather narrow in scope. Foundation models for biology along the lines of the LLMs that many of us are most familiar with are just now starting to be trained. One upshot of this is that we discuss different models for predicting shapes versus for predicting sequences. Obviously, in reality, these are two sides of the same coin, but for our purposes, they are often modeled separately. In the second hour, we discussed the implications of all this for human health, longevity, and biosecurity. I had heard, as you probably have, the story of how the first COVID vaccine was designed in just a few days after the right scientist received the required information, but I had never really considered what the world might look like if that pace of medical R&D were to become the norm. The impact would seem to be a near certain revolution, not just in biology, but also in practical medicine. One particularly striking theme is the potential for AI to change the way that we do biological research on humans. The difficulty and danger of experimenting directly on humans has always been a major bottleneck, but the latest models are quickly approaching the point where we should be able to run meaningful digital experiments. And that could radically accelerate the pace of discovery, both for the utopian better, and at least with some probability, perhaps catastrophically, for the worse. And all this, by the way, was before AlphaFold 3, which dropped shortly after we recorded, and which will certainly get its due attention in future episodes. With the full range of possibility in mind, and noting just how small many of these biology models are relative to the latest language models, I definitely want to give a shout out to the policy wonks at the White House who set a lower reporting threshold of 10 to the 23 flops for reporting on biological models as compared to 10 to the 26 flops for language models. That and their recent policy requiring DNA synthesis companies to screen orders against known dangerous sequences are looking extremely smart right now. By comparison, the now familiar question of whether today's chatbots are more helpful than Google for the purposes of making a bioweapon, to me, honestly, already feels quite quaint. For what it's worth, because the vision of the future sketched out here was so far outside my previous understanding, I did take some time to double-check my high-level interpretation of Amelie's claims with some very credible people in the field. One of my very smartest friends, who is also most deeply enmeshed in these issues, said simply, "That is exactly what is going to happen." If you find this work valuable, I would appreciate it if you'd take a moment to share it with friends. This episode in particular took a lot more work than a typical CEO interview, but it will be very well worth it if I can help our high-value audience get up to speed on such a critical area. And as always, we invite your feedback and suggestions either via our website, cognitiverevolution.ai, or by DMing me on your favorite social network. Now, I hope you enjoy this introductory deep dive into a genuinely awesome area of emerging research that may soon impact all of our lives. This is the AI revolution in biology with Amelie Schreiber. Amelie Schreiber, computational biochemist and AI researcher. Welcome to the Cognitive Revolution.
Nice to be here.
I'm excited for this conversation. I expect to learn a lot from it. This all started with a tweet that I saw you put out where you said, here are my top 10 AI tools for biology. And I read down the list and I was like, I don't know what any of these are. So yeah. I think pretty much immediately just say, hey, would you be interested in educating me in the form of a podcast? So I appreciate you taking the time. You want to start off by just giving a little bit of context on who you are, what you're doing day to day, and what kinds of problems you're trying to solve?
Yeah, sure. I'm an AI researcher, and I focus on computational biochemistry applications. I actually started out as a mathematician. My training in grad school was in mathematics, actually. Started off with pure mathematics and then transitioned into applied mathematics and data analysis. And then after grad school, I got into deep learning and started working on more AI related things. For me, the biochemistry applications are one of the most compelling things that we could be working on right now. I think other than like AGI, whatever that means, I think it's probably the most important problem we can be working on because it's a huge impact on human health. The applications are really profound and have the potential to be very impactful for everyone. So I get really excited about the biochemistry applications of AI. I have some projects and things that I'm working on that are more related to environmental things and material science type applications, but primarily I focus on the medical or biomedical aspect of things.
Okay, so let me try to give a extremely high level understanding of a couple of the biggest problems that are being studied in biomedical sciences. As I was preparing for this, I was having a good conversation with Claude 3 about it. And I came away with the understanding that both at the level of an individual cell and then again at the level of the overall organism system, we have one really massive challenge, which is that we don't know how it works. We've got the DNA, which is the code, the RNA, which is both messenger, but also is a machine. The proteins are all machines that fold up in weird ways and interact with each other in three-dimensional space. And then you've got the small molecules as well, which are like signaling, but also if they fit right, all these puzzle pieces fit together in super strange ways. There's this incredible network of interactions where things are disabling each other or promoting each other or interacting in all sorts of complicated ways. The nature of those interactions initially was entirely unclear. And now humanity at large has embarked on this grand project of trying to figure out how do cells work and how do our bodies work. And I'm gathering we're like maybe 5 to 10% of the way there. Most of the interactions remain unknown to us, but we've at least mapped out a decent chunk. So the first challenge, if you're trying to solve a disease, is figure out what is the pathway, what is the interaction in this like super complicated thing that is going wrong. And then the second challenge is, if I know what is going wrong, can I do something to intervene to stop it? But then again, there could be a lot of other knock-on effects. In that way, there's an important commonality with large language models. These things are not super clean. In language models, people are probably familiar with the notion of superposition, which is when activation in a network can light up for multiple different reasons. And we certainly see all of these kind of patterns of complexity in biology as well. But we know that there's a lot of stuff going on. There's a lot of interactions that are happening. We don't really know a lot about that, but we're gradually learning more all the time. And then if you do have a target, now it's okay. These are all three-dimensional spatial things. And so it's just extremely, extremely difficult.
It is, yeah.
How am I doing there in terms of just setting a foundation to understand the challenges?
I think you've really hit the nail on the head. I work with a lot of different kinds of molecules. So small molecules, which are your drugs that you like take in like a pill form. These are smaller than proteins and less complicated. And I also work with proteins and also a little bit with DNA and RNA. And understanding protein interaction networks is a very difficult and complex problem. To just determine whether or not two proteins interact with each other already is a hard problem. And then modeling how they fit together when they do interact and understanding the strength and how transient that interaction might be, it's very difficult. So protein interactions especially, but also protein DNA, protein RNA, protein small molecule interactions are all very complicated things to model. There are some new ways to address this that have come out recently that I have a lot of hope for, that I think are very promising, very effective approaches. I use various kinds of AI tools, analyze these molecules, create new ones, modify them, engineer them in ways, grafting them together and stuff, performing complicated operations on these molecules so that they perform a particular kind of function. As an example, designing a new protein to bind to another protein so that you can cause some kind of cascade event in a protein interaction network, or so that you can block certain interactions between proteins. A really good example in cancer, your basic first examples of a mechanism of cancer. There's PD-1 and PD-L1. These are two proteins. One of them's located in the cancer, and one of them's located on your immune cells. What happens is these two proteins end up binding to each other, and essentially the cancer turns off your immune cell so that your immune cell doesn't attack the cancer. And this is not good, right? You don't want this kind of interaction between these two proteins because you want your immune system to recognize the cancer and destroy it. Having this interaction turns off your immune system in a very specific way. So you can do things like design binders to this protein to block that interaction, and that can help you combat cancer and hopefully treat it. And that's just one basic example of something that you can do with these tools that I'm using and that I'm interested in. You can also design new drugs with them. You can design drugs that are specific to a particular binding pocket on a protein. You can design drugs that have very specific chemical properties. You can design proteins that have very specific chemical and functional properties. And this is pretty new stuff, but you can also do a lot of the same things with DNA and RNA molecules as well.
Okay, this is a paradigm shift, right? The application of AI to biology, obviously AI is creating all sorts of paradigm shifts. But I think it might also be helpful for people to understand a little bit better the before state when we didn't have any of these tools yet, which is not that long ago.
Not that long ago, yeah.
What was the sort of prevailing approach to figuring stuff out? You hear these stories of, oh, look, we found this frog in the rainforest that is immune to a certain disease. What's going on there? Let's see if we can't find something in this frog that could be a medicine or whatever. It's really anecdotally special observation motivated investigations in a lot of cases. And then I know there's also just a lot of brute forcing where it's like, we have no idea which proteins are going to interact with which other ones. So let's just create this massive cross matrix and see if we can't figure it out that way and look for hits, kind of massive assays, just exploring the space. All of these things without any idea of what the puzzle pieces actually look like, which makes it obviously very difficult to figure out how they would fit together. What more would you tell people who want to understand, okay, what was the before all this stuff started to come online?
There's a lot of methods that come from wet lab work where people do this stuff in animals. Like they say you want to do directed evolution on a protein and try to find higher functioning variants or variants that are more thermostable, that have higher expression or something like that. You can mutate these things in a lab. You can do like point mutations, or you can do 2 mutations at a time, or you can do multiple. But when you start adding in things higher than just single point mutations, you get this combinatorial complexity, right? And so it gets really unwieldy. I feel like my experience with traditional methods is somewhat limited. I don't come from a wet lab background. I come from a very computational background. I haven't spent a lot of time working with more traditional methods that people have used historically. So computationally, traditionally, the way that this sort of thing was approached was through molecular dynamic simulations. You have what's called a potential, and this potential tells you how the dynamics evolve.
And is that basically like a energy potential diagram? Help me pin down the ground truth.
Yeah, observing the dynamics of a protein is really hard. The ground truth ultimately is the Boltzmann distribution, which is this theoretical physics idea that comes from statistical mechanics. It's a probability distribution. And you can think of it in terms of an energy landscape. The low parts, the valleys that you have in your energy landscape are going to be the metastable states of the protein. And then the peaks, like on top of the mountains, right? These are states that are very transient and that are unlikely to exist for very long. So when you're trying to model the Boltzmann distribution, you're trying to get out these low energy conformations from it, and then the transitions between those, and understanding how often you transition between this state and this other state and what that transition looks like. This is all part of the dynamics of the protein in the environment. And so I guess the answer to your question is the ground truth is the Boltzmann distribution. Whether or not you can model that on a computer or observe that in a lab is another question. But the Boltzmann distribution of the protein is the ideal ground truth.
So it's a probability distribution of what percentage of the time the protein in some solution or whatever is in shape A versus shape B versus shape C. So in terms of just a little intuition for the shapes, I envision a slinky where if I just sit the slinky down on the floor, it will come into a pretty tight coil. And that you might say is like its lowest energy state. Then I can stretch it out. And if I, do the work and put the energy into it, then I can stretch it out. Now, if I let it go, it's going to snap back to its low energy state. That reverb moment in between things is a transitional state. It's not going to be in that state very long. It's on its way from one to another.
That's right, yeah.
And for any given protein, there could be multiple different low energy states that it might spend time in.
Yeah.
And of course, the individual proteins, they're in solution, right? So there's water molecules bouncing off them all the time. So the constant bumping into the environment creates opportunity for these things to occasionally flop from shape to shape.
Yeah.
And there probably also is like a path issue where you maybe can get from A to B and B to C, but not necessarily A to C without going through B, I imagine.
Yeah, exactly.
And so doing all of this is probably hard for a lot of reasons, but you highlighted the fact that the computation is really slow, right?
Yeah, so molecular dynamics simulations, they come in a lot of different flavors, a lot of different complexities. Some of them model the proteins using just standard Newtonian physics, and then you can add in more complexity on top of that, things like quantum properties and other features of the protein to make the molecular dynamics simulation more complex and more robust. But these simulations are really computationally intensive. They take a long time, they take a lot of GPUs, and they're just not very efficient. And on top of that, the length of time that you run your simulation for really, in a lot of cases, determines how accurate your distribution is and how accurate the conformations or trajectories that you get out of that are. So you might run your simulation and not run it for long enough, and you don't get all the different states that the protein might be in, but you miss some of them, right? And there are some new AI models that are trying to address this and trying to make headway into augmenting or even replacing molecular dynamic simulations in a lot of cases. So for example, AlphaFold 2 came out a couple of years ago, and that was a big deal, right? But you just get a single static structure from AlphaFold 2. So we give it the protein sequence, which is just a sequence of amino acids, which are represented by 20 letters. And it takes in this protein sequence, and it provides you with a static structure for that protein that's a low energy conformation for that protein. It also tells you how confident the model is that that is the structure of the protein at that particular point in the protein. And then you can take the average and get an overall confidence for the whole protein.
Hey, we'll continue our interview in a moment after a word from our sponsors. AI might be the most important new computer technology ever. It's storming every industry, and literally billions of dollars are being invested. So buckle up. The problem is that AI needs a lot of speed and processing power. So how do you compete without costs spiraling out of control? It's time to upgrade to the next generation of the cloud, Oracle Cloud Infrastructure, or OCI. OCI is a single platform for your infrastructure, database, application development, and AI needs. OCI has 4 to 8 times the bandwidth of other clouds, offers one consistent price instead of variable regional pricing, and of course, nobody does data better than Oracle. So now you can train your AI models at twice the speed and less than half the cost of other clouds. If you want to do more and spend less like Uber, 8x8, and Databricks Mosaic. Take a free test drive of OCI at oracle.com/cognitive. That's oracle.com/cognitive. oracle.com/cognitive. The Brave Search API brings affordable developer access to the Brave Search Index, an independent index of the web with over 20 billion web pages. So what makes the Brave Search Index stand out? 1, it's entirely independent and built from scratch. That means no big tech biases or extortionate prices. 2, it's built on real page visits from actual humans, collected anonymously, of course, which filters out tons of junk data. And 3, the index is refreshed with tens of millions of pages daily, so it always has accurate, up-to-date information. The Brave Search API can be used to assemble a data set to train your AI models and help with retrieval augmentation at the time of inference, all while remaining affordable with developer-first pricing. Integrating the Brave Search API into your workflow translates to more ethical data sourcing and more human representative data sets. Try the Brave Search API for free for up to 2,000 queries per month at brave.com/API.
So let me just get into a little bit more how that happens. If I understand correctly, there were proteins that had been analyzed mostly through X-ray crystallography to the point where people were pretty confident that they had a structure. People put a lot of time in the lab. We got to make enough of this stuff, get it to coalesce into some crystal form. Then we can hit it with X-rays. Then we can try to decipher how that gets scattered. And then we can come up with a structure. And we hope that approximates the structure that it actually takes in the cell. But no guarantees, because protein crystals don't really occur in nature, right? That's very an odd thing in the first place. So that was like closest approximation that we could get, right? So that, I think that's why the initial things were limited to outputting a single predicted structure.
That's right. But again, that protein could exist in other conformations. It might have other states that it exists in when it's interacting with other proteins, or based on the environment that it's in, what temperature it is. Things like this can change the shape of the protein, right? They move around. They're very jiggly, and they do things. And so having a way to sample the Boltzmann distribution and get all of these different conformations out of them, and also understanding the transitions between these states, is a really difficult problem. People have been trying to address this. There's actually a model that just came out middle of last year that does a really good job of addressing this. It's called distributional graph former. It's a generalization of AlphaFold 2. It's not just a single static structure anymore. It's actually a whole ensemble of structures, and also the transition pathways between those different metastable states. Distributional graph former does a pretty good job of doing it too. There's some room for improvement for sure, but it's a pretty solid model. Some people that I have a lot of respect for worked on this, and it's a diffusion model similar to like DALL-E, which most of your watchers are probably familiar with, but it works on proteins instead of images.
And then another level of like insane complexity. They interact with each other. So this is like maybe in the presence of some other protein that constrains it in a certain way, or some other small molecule perhaps that fits into a pocket of it in a certain way. You can have these deformations, and then subject to those constraints, they still find their natural low energy state. Returning to my slinky visualization, I could also step on the middle of it, and then the parts on the side would presumably still look like normal, but there'd be this like deformed part in the middle where I'm stepping on it. It's where AlphaFold multimer starts to come in.
Yeah. AlphaFold multimer models the interactions between proteins. When it predicts the two proteins together, there's an interface region where the proteins are in contact with each other. And AlphaFold multimer tells you the quality of the interfaces between the two proteins. And then you have these predicted aligned error outputs. And these tell you how good of a quality the model has provided you.
So on the interface, you have a range of kind of whether these things are like puzzle pieces that are perfectly fit to each other and have a really tight coupling, or jigsaw pieces that sort of fit, but they don't perfectly fit and they don't really want to stay together as much, all the way down to obviously they just don't fit at all and they just kind of don't adhere to each other.
Yeah.
Is there anything more we could do to develop our intuition there for this dynamic range of how adherent these things are to each other?
Yeah, so there's two ideas that you're addressing here. One of them is binding affinity, like how much affinity these two things have for each other to bind to each other. And then there's also how transient are the interactions between them in terms of like dynamics and like the time spent next to each other. There's recent work, I think it's out of MIT. They used the AlphaFold 2 architecture and they train it as a flow matching model, which is a generalization of diffusion. And using this, they get these ensembles of conformations for a protein. And part of the information that they get out of this is how transient the interactions between residues are. Let's say you run your AlphaFlow model and you produce 10,000 different conformations or states that the protein exists in. You can cluster those structurally and then look at which residues are close to each other in each of those clusters. And you can tell how transient the interaction between these two residues are and how much time they spend together.
One thing that caught my attention, I believe you had said that there were like as many as 10,000 possible conformations for a single thing.
Yeah, when you run AlphaFlow, it generates conformations of a protein for you. And it was trained on molecular dynamics simulation data. So it does have some notion of dynamics. And it produces different conformations of the protein for you. And you can tell it how many of these to produce, right? And the more that it produces, the more likely you are to get a nice global picture of all the different conformations that might exist for that protein. So we've actually tested AlphaFlow on proteins called fold switching proteins. The most popular example in the literature and in the community right now is Chi-B. And Chi-B is this fold switching protein that like 10% of the time it exists in this one conformation, and then 90% of the time it exists in this other conformation. And this is influenced by like a circadian rhythm. And when you apply AlphaFlow to some of these fold switching proteins, it doesn't capture both of those fold switch states. So in the example of Chi-B, it actually only really predicts conformations that are relatively close to the slightly higher energy conformation, the one that's a little bit less likely, the one that it exists in 10% of the time, which is a little bit odd, right? Because you would expect and maybe want your model to go for the ground state or the lowest energy conformation. And for some reason, it sticks kind of close to the fold switch conformation. I'm not sure that there's like a good explanation for why. I think it goes back to the AlphaFold 2 predictions, because when you predict the structure using AlphaFold 2, you get the fold switch state, you don't get the ground state. People have done all kinds of little hacks to try and get out the ground state instead of the fold switch state. The most popular method they've hacked together is doing MSA subsampling.
Can we talk about this MSA thing for a second?
MSAs are basically this. A lot of times, the changes that happen only happen in certain regions of the protein, and then there are other regions of the protein that are highly conserved. And so when you do an MSA, you can look at which regions of the protein are really highly conserved. And this will give you some indication of which parts of the protein are going to affect the function if you mutate them. So like if I have the region of the protein that evolution hasn't changed or has changed very, very infrequently, if I change that, if I mutate that, it's probably going to degrade the function of the protein or change the function of the protein, or maybe you'll decrease its thermostability or something like that. So MSAs are just telling you evolutionary information, and AlphaFold 2 uses MSAs. You can either use the database of MSAs or you can have it compute them on the fly to inform how it predicts the structure of the protein. Because when you have proteins that are very closely related evolutionarily, their structures are often very similar, right? That's not always true. Like I can have two proteins that are very similar but have very different structures. Like that happens all the time. But if you have this extra evolutionary information in the form of an MSA, you have extra information that helps you predict the structure of the protein of interest.
This is sort of calling to mind the classic image of the plane with all of the spots where the planes came back having been shot, and then all the places that they didn't observe, those were the planes that got shot down. So this is the evolutionary equivalent of that, where we don't see changes in certain regions because they're super core to functionality. And that in turn is super useful for prediction making because these parts are the really important parts, and the sort of interactions that they have are the whole point of what's going on.
And so clustering the MSA captures certain evolutionary information. Different clusters will focus on or accent particular conformations. And so if I take out one of my clusters and predict the structure using those clusters in the MSA, I'll get a conformation out from AlphaFold 2. And then if I pick a different cluster of sequences in my MSA, I'll get maybe a slightly different conformation or maybe a drastically different conformation. And if you do this right, sometimes you can get out the different conformations of these fold switching proteins. And for Chi-B, they've done this successfully, and for a few others, but it's not a super robust method. And I think AlphaFlow is more informative. Boltzmann generators or distributional graph former, I think, are significantly better in terms of how they approach the problem because they're going to give you more robust information.
Okay, that's interesting.
So yeah, I think AlphaFold multimer works really well. You give it the two proteins or multiple proteins, and you find out if there's an interaction there. It tells you what the quality of the interfaces are between the two proteins that you're predicting in the multimer. You could generalize this, right? And this is something that hasn't been done yet. So people who are looking for like research projects, this would probably be a pretty good one. If you generalize this to AlphaFold multimer, do the AlphaFlow thing with AlphaFold multimer, if it works well, you could figure out how transient those interactions between two proteins are, right? Because now you have two proteins or more that are in different conformations at different times, and AlphaFlow will generate all these different conformations for you, and then you can analyze all these different conformations and figure out how transient the interactions between the two proteins are and get an idea of the dynamic side of things. So hopefully that gives you some idea. I think computational approaches are proving to be much faster, much more effective, and you can scale them. And this is really good because we need things that are more computationally efficient so that we can do this for a lot of proteins, right? Because we have a lot of proteins to do this for. We're working sometimes with millions or hundreds of millions of proteins. Having millions of variants of a protein and being able to assess them and determine their quality in some metric has become a lot easier in the past couple of years even. So like a good example of how AI has sort of replaced wet lab methods, doing directed evolution in a lab in a lot of ways, now that we have AI tools to do similar things, feels a little bit unnecessary. Like, why do we need to go and inject an animal and wait some amount of time for this to play out inside the animal and then actually synthesize these things by hand in a lab somewhere when we can like do very similar things computationally and often get better results?
Hey, we'll continue our interview in a moment after a word from our sponsors. Hey all, Eric Torenberg here. I'm hearing more and more that founders want to get profitable and do more with less, especially with engineering. Listen, I love your 30-year-old ex-FANG senior software engineer as much as the next guy, but honestly, I can't afford them anymore. Founders everywhere are trying to turn to global talent, but boy, is it a hassle to do at scale, from sourcing to interviewing to on-the-ground operations and management. That's why I teamed up with Sean Lanahan, who's been building engineering teams in Vietnam at a very high level for over 5 years to help you access global engineering without the headache. Squad, Sean's new company, takes care of sourcing, legal compliance, and local HR for global talent so you don't have to. With teams across Asia and South America, we can cover you no matter which time zone you operate in. Their engineers follow your process and use your tools. They work with React, Next.js, or your favorite front-end frameworks. And on the back end, they're experts at Node, Python, Java, and anything under the sun. Full disclosure, it's going to cost more than the random person you found on Upwork that's doing 2 hours of work per week but billing you for 40. But you'll get premium quality at a fraction of the typical cost. Our engineers are vetted top 1% talent and actually working hard for you every day. Increase your velocity without amping up burn. Head to choosesquad.com and mention Turpentine to skip the waitlist.
Omneky uses generative AI to enable you to launch hundreds of thousands of ad iterations that actually work, customized across all platforms with a click of a button. I believe in Omneky so much that I invested in it, and I recommend you use it too. Use CogRev to get a 10% discount.
So it seems in some sense surprising that you would be able to make a new black box system to make these predictions that they would be faster and more accurate as opposed to just running the physics. I have a hypothesis on this, but how is that leap happening?
Okay, so I think the key here is compression. Neural networks are compressors of information. Whereas in molecular dynamics, you could simplify things by simplifying the forces or simplifying the model in terms of how complicated the physics are that you're using to model problems. So like if I strip everything down and just do like bare bones Newtonian physics, I can simplify things that way, but there's no real compression happening. For these AI models, you could think of them as functions, but you can also think of them as compressors of information. You're taking something complicated and noisy sometimes, and you're compressing it, and you're providing a representation of it that is more compact. Traditionally, you think of a deep learning neural network, you've got data, you train on your data. Maybe you have a train test validation splitter. You train on your training data and you see how it performs on your test data, and that's your trained model. But you can also train models with physics constraints, right? There are some approaches that people are using that are completely data-free and are based on physics. And the model is learning the physics and compressing that physics. You get something that is faster, like orders of magnitude faster, right? You get something that produces your answer in like a minute instead of 4 hours or days. And if you've done it right, it generalizes to systems that it hasn't been trained on before. And people are doing this for this problem in particular, for getting the Boltzmann distribution of a protein and getting all these ensembles of conformations and their transitions between the states. Does that make sense?
Yeah, that was pretty much my hypothesis, that it's essentially learning higher order concepts beyond the raw physics. This striking observation that comes up over and over again that, okay, yeah, language models, they're only trained to predict the next token, but they not only seem to be generalizing certainly beyond the narrowly defined bounds of their training data, and perhaps to some degree even more than that, a hotly debated topic. But what is pretty clearly demonstrated at this point is that in the middle to late layers of a language model transformer, the techniques are there to say, okay, this pattern of activations seems to correspond to this higher order concept that we care about, which is kind of a miraculous thing, that it's just predicting the next token, but it's learning these concepts of justice and fairness and ethics and whatever that are obviously useful to predict the next token. And that's presumably why they're arising, but not something that's been like specifically coded for. So I guess in the application to biology, basically the same phenomenon is happening, where raw data or raw simulated physics or whatever is the input. I'm guessing that the token level vocabulary of a protein would be the 20 amino acids, but the sort of higher order concepts are like, oh, this chunk of a thing is reused a lot. Yeah. And these two chunks of things interact with each other in a particular way. So give us a little more intuition of that. What are the higher order concepts that these things seem to be learning?
Yeah, so the concept that I think you're trying to grasp onto is protein motifs. Motifs are like reoccurring patterns that happen in proteins. They're short little sequences of amino acids that recur often in lots of different proteins and generally have a very similar structure across different proteins. If we put aside dynamics for a moment and we just look at like structure prediction, so there's another model, ESMFold. This is an alternative model to AlphaFold 2 that does essentially the same thing. It predicts the 3D structure of the protein in some low energy state, right? It doesn't perform as well as AlphaFold 2, but it does pretty well. And it's a language model. It's something that people call a protein language model. It's built on the BERT architecture, actually, which sounds kind of bad, right? Because BERT's like this older model that's only used for like specific things now. The ChatGPT models have overshadowed or outshined the BERT models at this point. But in biology, this actually makes a lot more sense because you have the masked language modeling objective that you train on for protein sequences, and you just mask out some of the amino acids in the protein sequence and have it predict what those are, right? And just by training it to do this, and then putting a folding model on top of it called evoformer, which actually comes from the AlphaFold 2 architecture, you don't train on any physics, but somehow you learn how to predict 3D structures of proteins. And so in this case, it's almost like physics mostly isn't needed. If you just want to predict a static structure of a protein, you can get pretty good results just using a BERT type architecture with mask language modeling objective, training on millions of proteins. You get something that will fold proteins for you pretty darn well. There's some really nuanced architectural differences. AlphaFold 2 uses multiple sequence alignments, and it also uses templates. And in general, it is better performing because it has these extra added things in the architecture that improve its ability to predict the structure. But even there isn't really a lot of physics explicitly happening. We're not giving it forces and potentials and things like that. And yet somehow we're able to predict the structure of the protein with really high accuracy for a lot of proteins, for most proteins. I guess that's another example of where AI can be a lot better than the molecular dynamics, because if you want to model a protein actually folding in a molecular dynamics simulation to get the folded structure of the protein, this is pretty hard and time consuming and computationally intensive. Whereas for AlphaFold 2 or ESM fold, you give it a big protein and it takes a few minutes. These are language model-like models, especially ESM fold. ESM fold is pretty much just a language model. And so they are learning higher order concepts for sure, just as they do for natural language. You can do topic modeling and things like that on these models where you're looking at groups of amino acids instead of just the individual vocabulary elements. Another thing people have found is the attention maps recapitulate the contact maps for proteins. The contact map is like a 2D matrix representation of all the contacts between the different amino acids in the protein. And it turns out the attention map, the matrix that you get from your attention mechanism, recapitulates that and is highly correlated with those contact maps. There's some work on this, maybe 3 years old now, called Biotechnology meets biology. And they do a really in-depth study of how to pull out these different things, active sites and binding sites and motifs and things like this, based on the attention maps in the protein language model. But I would say training a model to predict binding affinity is a hard thing to do. And there's actually a big problem with a lot of these models. Most models that try to predict protein interactions don't generalize well. When people are training them, a lot of times people don't split their data in the right way. If your training data has sequences in it that are very similar to your test data sequences, you're going to get overfitting and you're not even going to realize it. And a lot of the protein interaction models that have been trained don't take this into account, and they don't split their data based on sequence similarity or structural similarity.
The overfitting piece, I didn't quite understand it.
So when you're training your neural network, you want to make sure that your test data doesn't have a bunch of sequences in it that are highly similar to your training data, because it's almost like you're training on the same thing twice. And you overfit this way, you don't get a good indication of how well your model generalizes. And a lot of people do this. A lot of people coming from deep learning, they don't have a super strong background in biology, they make this mistake very often. And so there's just tons and tons of models out there that don't generalize and that are very overfit because they didn't split their data based on sequence similarity and or structure similarity.
Interesting. Okay.
Yeah.
That's not really done in language modeling, right? In language modeling, you could certainly have lots of sentences that are very similar, and you don't draw conceptual lines between different parts of the data and try to generalize across those lines. So what is the difference in the biology context? My intuition says, to the degree that these models are learning the higher order concepts that really matter, that they should be able to learn those kind of regardless. And again, in the language model case, we don't really have a concept of overfitting, at least in the large scale foundation models. Is this?
I mean, there's still a concept of overfitting in NLP and training big giant models, Claude or ChatGPT or whatever. You can definitely still overfit these models for these really big chat type models, like really big LLMs and stuff. First of all, they often only train on one pass through the data, right? They don't do multiple passes. And so partially because of that, overfitting is not much of an issue because you're not training multiple times on the same data.
So I do think that is changing. One little nugget from the recent Zuckerberg-Dwarkesh interview that I definitely made my ears and brain light up was when he said they trained these latest llama models on 15 trillion tokens. He said we stopped because at some point we need to move on to Llama 4. But he said we could have rotated the high value tokens through again. And
Sure.
I was like, oh, interesting.
Yeah.
Anyway, that's a bit of an aside, but I'm still
yeah.
not quite grokking it, to be honest.
Okay. So let's see. So you have your training data and your test data. Like your test data is supposed to tell you something about how well the model generalizes, right? If the metrics on your test data are substantially worse than the metrics on your training data, then you've overfit, right? So to get a good idea of how well your model is generalizing to unseen data, you want at least part of your test data to be very dissimilar from your training data. And this is a big thing that people discuss, is like whether or not these things generalize to data that's out of distribution. And if you train them in the right way, you can get models that generalize well to out of distribution data. That means it's picked up on some deeper concept that generalizes to areas that it hasn't seen before, and it's using those really deep concepts to make predictions or generate things that are out of distribution. This is not something that's impossible to do. You just have to do it right. And in the case of biological sequences like proteins, the way that you do this is you look at the sequence similarity between the protein sequences in your training data and your test data. And you can also look at structural similarity, because you can have different sequences that fold into the exact same structure, more or less. And so sometimes, depending on what your model architecture is and what your goal is, you may also want to split based on structural similarity and not just sequence similarity, to make sure that your model is generalizing and not overfitting. Does that make sense? Yeah.
I think so. For any given architecture, presumably the very best performance would be if you trained on all the data. But
sure,
in order to effectively evaluate your architectures along the way, you need to have some holdout to evaluate against. And these similar enough sequences basically are a data leak, in the same way that if you find out after you do your thing that, oh, hey, we actually had MMLU in the training data for the language model, then it means like, oh, well, you can't really trust the MMLU scores anymore.
Right.
And the model still may be good or may not be so good, but if you train on MMLU, we can't really score you on MMLU. And this is a similar type problem.
Yeah. Exactly.
Okay.
And there are even more nuanced ways of splitting the data too for specific things. They trained a new version of DiffDoc. I think they call it DiffDoc L, because it's larger. It's the large DiffDoc. And they use this method called confidence bootstrapping, which is sort of like reinforcement learning. And they also split their data in a really unique way. They split their data based on these domains that are present in these interactions to train a model for docking. So you have a ligand, a small molecule ligand that you're trying to dock into a protein and do like blind docking, which is a hard problem. And they get like substantially better performance, and it generalizes a lot better in this new version of DiffDoc because of the way that they trained it and the way that they split their data. And they have really strong confidence that their model is generalizing to out of distribution data, and it's able to predict on things that it hasn't seen before and that are very dissimilar from what it has seen.
So just to make sure I understand, what they've demonstrated is that you should be more confident in doing this stuff in totally new regimes based on the fact that they've been very meticulous in the data split. Whereas if they had thrown everything into the training, then you would have a hard time knowing to what degree to be confident in out of distribution stuff. Yeah. Okay, cool.
And they did a really good job with that model. I think their performance boost was pretty big. They got a lot better performance out of it too. So it's definitely a good model to look into if you're looking to get into this stuff. Something else we should probably get into is training models that are like AlphaFold that do more than just proteins. You might have complexes where there's a protein in a small molecule, or protein in DNA, or protein in RNA, or a metal, or whatever. There's all these other biomolecules that you might want to model in complex with each other. And to generalize AlphaFold multimer to this new situation is pretty hard, but people have managed to do a pretty reasonable job of it. There's still room for improvement on these, but A new model that just came out recently is RosettaFold All Atom. RosettaFold All Atom will let you predict complexes that have proteins and small molecules, proteins and nucleic acids, and proteins and metals too. Big step forward now, because now you can apply this idea to these other complexes. And you can get this score that tells you how likely there is to be an interaction there and how strong that interaction is. And you could also probably do the AlphaFlow type method with something like RosettaFold All Atom as well, and get something like conformational ensembles out of it. And that something like that is probably coming soon. So yeah, I would say now a lot of the more interesting models are more influenced by diffusion and flow matching models. A lot of the generative models that we're getting that are predicting the Boltzmann distribution, or that are generating new protein structures for you with specific shapes and functions, or that are allowing you to design new sequences that fold into a particular backbone. A lot of them are more influenced by diffusion and flow matching than language modeling and transformers. So some of them use transformers, but they're starting to be a lot more influenced by DALI type models, I would say. You're starting to get this really fine-grained control over what kinds of proteins or small molecules or nucleic acids that you can generate. And there's actually some models that have come out that are text conditioned. So a couple of the models that I mentioned were protein DT and molecule STM. These are generative models that are text conditioned. They use the exact same method as CLIP. They use contrastive learning, and they have captions and molecules or captions and proteins. And they allow you to type in a natural language prompt and get out a molecule. So protein DT, for example, or molecule STM, you can give it a text prompt in natural language describing the properties of the molecule in just natural human language, giving it this molecule has these chemical properties and it has this sort of bias away from or towards these amino acids, or interacts with these other molecules in such way. You can give it these natural language text prompts and it will generate proteins and small molecules for you that satisfy these constraints, or that correspond to the text prompt that you give it. And there are more models like this that have come out recently that do similar things. They're text conditioned, diffusion models or flow matching models that are generative and that produce molecules with specified properties based on natural language text, which in my mind, that's kind of mind-blowing. I think that's amazing. Having a model that will just generate molecules for you that fit natural language descriptions, that's crazy. That's amazing.
Yeah, no doubt. Okay, in actual work, what are the cycles that are used to actually make progress on questions of interest? You have a target, you want to make some modification. How do these things fit together to allow us to work a lot faster and get more value from our limited wet work resources?
Are you familiar with RF diffusion at all?
No, not really.
RF diffusion is a diffusion model that used the RosettaFold backbone and trained it as a diffusion model to denoise 3D structures of proteins. You don't give it a text input, but you can condition it on other types of things. It's a little more specific, and it's a little more geared towards people who want to perform surgery on proteins. RF diffusion doesn't usually get used on its own, because RF diffusion just works at the level of structure. It's doing the denoising process on the structures of the proteins. It doesn't know anything about the sequence of the protein. So I need a separate model like Ligand and PNN to design the sequence for that 3D structure. So they kind of go hand in hand. RF diffusion designs the structure, and then Ligand and PNN designs the sequence for that structure that will fold into it, so that you have something you can actually go and synthesize in a lab, right? You need a protein sequence to synthesize to get the protein structure. So once you've generated your 3D backbone of your protein, you design a sequence for it using Ligand and PNN. So if you want to design new proteins with specific structural properties, and you want to get your hands dirty and perform surgery on these proteins and graft them together, or take pieces from this one and graft it onto this one, or generate a protein with a specific sort of fold tertiary structure, or maybe generate a protein that binds to a specific protein with really high affinity and specificity, These are the things that RF diffusion is good for. There are several different versions of RF diffusion. There's a new version that just came out called RF diffusion all atom, which allows you to generate proteins that will bind to small molecules. So you can generate proteins with binding pockets that are highly shaped complementary to a small molecule ligand. And with RF diffusion and RF diffusion all atom, you can also do something called motif scaffolding, which is where you pull out motifs from a protein that you like. Maybe they're like binding sites or active sites, or maybe there's a particular segment of the protein that binds to a different protein that you're interested in or performs a specific function. And you can pull these pieces of the protein out and scaffold them and build a new protein around those pieces that holds those pieces in place, so that you have a new protein with very specific 3D structure that performs a very specific function. It's been a very influential model and a very useful model, and it's definitely one of my favorites.
What exactly it is that you're specifying? You're saying, I want it to have certain properties, and then it's filling in the sequence that gets you to those properties?
Yeah, the most basic usage of RF diffusion is unconditional generation of a protein. So you tell it some length or range of lengths, and it will just generate proteins of that length that are brand new, that have never been seen in nature before, and very often are very designable and that you can actually synthesize and create in a lab. And that's just completely unconditional generation. You don't give it any sort of constraints. It just generates a new protein for you of a particular length. One of the really interesting functions of RF diffusion is it will design binders for you. So if I have a target protein and I want to design a protein that binds to it with really high affinity and really high specificity, RF diffusion is really good at that. It'll design binders for pretty much any protein you can give it. And very often, the proteins that it designs that bind to your target protein are very high affinity and very high specificity, and often very thermostable. And using ligand and PNN, you can increase your binding affinity and other properties even more. And then the other functionality that's really useful is the motif scaffolding, which is where you pick out pieces of a protein or multiple proteins, and you build a new protein that holds all those different pieces in place in a particular way. And so one thing that you could imagine doing with this would be like building or designing an adjuvant, or like grafting two proteins together. Or let's say I have one part of a protein that binds to protein A, and I have some other protein that binds to protein B. And I want to design a protein that binds to both protein A and protein B, so that I can get a complex with three proteins in it, right? I want to have a protein that binds to protein A and protein B. So what I can do is I can take those pieces out of the protein that bind to protein A, and I can take the pieces that bind to protein B, and I can scaffold them together into a new protein. And now I've got a new protein that binds to both of those proteins. And I can do things like build out protein interaction networks this way. So if I want to design a particular protein interaction network, this is really useful for that. That's a pretty technical and involved thing to do, is a non-trivial thing to do, but you can do it with RF diffusion. You can design proteins that will bind to multiple different proteins in multiple different contexts and build out these networks this way. And this gives you like a way to modulate protein interaction networks, right? Because you can modulate your protein interaction network by blocking particular interactions by designing a binder, but you can also add in interactions that weren't there before using the motif scaffolding, or maybe binder design and motif scaffolding together. So it's very much geared towards people who are like really interested in performing surgery on proteins and protein interaction networks. And it's very useful in a lot of different contexts. And now with RF diffusion all atom, also with other interactions as well, because with the all atom models, you can model more interactions than just the protein interactions.
So how does this in practice get used? That maybe the cancer example from the top is a good one.
Yeah.
In that scenario, we already had an understanding of the cancer cell has this thing that disables the immune cell. And so then you could say, okay, let's brainstorm some ideas here, right? If we can bind something to that bit of the cancer cell that would blunt its ability to then interfere with the immune cell, then the immune cell presumably would do its job still and kill the cancer. Of course, you've got highly tangled webs of interaction that you're doing this within, but you've got something local like that. Now, kind of take me through the cycles and the practical application of some of these tools for a specific problem of that sort.
Yeah, so like the one that we were talking about earlier with the cancer, with PD-1 and PD-L1 is a really good first example that I think is a really good place for pretty much anybody to start, because you can very easily and very quickly design a binder to one of these proteins with RF diffusion. It's really good at designing binders for pretty much anything. If I have this PD-1, PD-L1 interaction between these two proteins, essentially what's happening is the cancer is turning off your immune system's response to it, and then your immune system doesn't effectively combat the cancer. But if I design a binder to one of these two proteins to block that interaction, and my binder outcompete that interaction, so it has a higher affinity than the PD-1, PD-L1 interaction has, then I can successfully block that interaction and prevent the cancer from turning off the immune cell's responses to it. Another way that RF diffusion can be used, so let's say you have a protein that binds to a target, but it doesn't bind very well. The affinity is low and the shape complementarity isn't terribly good. I can use RF diffusion and protein MPNN or ligand MPNN to modify that and increase the binding affinity. So something I can do with RF diffusion is partial diffusion. So I can take my protein of interest that binds to my target and I can perform partial diffusion, which is where I add a little bit of noise. I don't completely noise it. I don't turn it into a Gaussian distribution or anything. I just add a little bit of noise into it and then have RF diffusion denoise that. And what it'll do is it'll denoise it into a structure that's kind of similar to the one that I started with, but it's also a little bit different now because I added that noise and then denoised it with RF diffusion. And if I do this and I give RF diffusion the target protein that I'm interested in, I can improve the shape complementarity between those two. So the new denoised protein, or the new denoised version of my original protein, is going to have a higher shape complementarity to my target. And then I can go and design sequences for that backbone that are different from my starting sequence, right? So maybe my starting sequence, maybe there were some residues that had unfavorable chemical properties at the interface, right? Maybe I need to use different amino acids to improve some chemical properties so that the binding affinity goes up, right? I can do that with like protein MPNN or ligand MPNN. So ligand MPNN is the newer all-atom version of protein MPNN. It's an improvement on protein MPNN, and it also generalizes to contexts where you have other kinds of molecules other than just proteins. And I design a sequence with ligand MPNN, and I can tell ligand MPNN to bias certain residues towards certain amino acids or away from certain amino acids, like at the interface, for example, to get those more favorable chemical properties to increase my binding affinity further. There's also versions of ligand MPNN that will allow you to specify whether a residue is a buried residue, an interface residue, or something else. Yeah, so like using partial diffusion with RF diffusion to slightly noise and then denoise a structure that you already have can help you increase like things like binding affinity and things like thermostability and other properties. Then let's see, what's another application of RF diffusion? Okay, symmetric generation. You can design symmetric oligomers. Let's say I want to have a protein that interacts with other copies of itself. And if I have enough copies of this protein, it forms a symmetric complex. For example, let's say I have a protein that fits together with two other copies of itself in a triangle formation, right? And it's symmetric to the cyclic group of order 3, right? So I've got a order 3 rotational symmetry that's happening. I can use RF diffusion to generate proteins with symmetry like this. And it has other symmetries that it can use. So there's dihedral symmetries, there's symmetries icosahedral symmetries and tetrahedral symmetries, in addition to the cyclic symmetries. And the cyclic and the dihedral, I can choose like any order cyclic group or dihedral group for my symmetry for those. And RF diffusion will design a symmetric complex of proteins where there's like multiple copies of the protein in this like nice symmetric structure. This occurs in nature in multiple places. One example is in viral capsids. And they've also recently, David Baker's lab recently published some work where they designed these like symmetric oligomers as like biosensors. And I think they also designed some to help with like drug delivery. So they have these like the like pocket that forms when you have a cyclically symmetric complex of proteins, the pocket that forms inside of that, you can design that in such a way that it captures a small molecule drug really well and delivers it to somewhere in particular, right? So that's another application of RF diffusion that's really useful. What else? There's the binder design, fold conditioning, which is where you can tell it like specific tertiary structures to condition on. Maybe I want to design something like a TIM barrel. I can tell RF diffusion to generate something that has the rough 3D shape of a barrel. So I can tell RF diffusion specific tertiary structures to generate, and it will generate these different folds for you. It's called fold conditioning. So here's a good example of how to use motif scaffolding. Let's say if I want to design an inhibitor for two different proteins that are interacting with each other, I could extract the motif corresponding to the interaction and then use that to design a new protein, scaffold that with the motif scaffolding. And then I can optimize it using like partial diffusion and like ligand MPNN to design sequences that have like favorable chemical properties. And then I can check and see using the LIS score from AlphaFold multimer how well my new protein interacts.
If I'm getting it, it's like, okay, we have an interaction that is problematic. We want to interfere with it one way or another. We can like cap the one thing or cap the other thing. And so there's this kind of cycle of generate a 3D structure. That's one model. Generate a sequence to do that. That's another model. Refine those to look for favorable chemical properties. And then you're validating this to the degree that you can validate it before you're actually doing any actual lab work with something like AlphaFold multimer and looking for a high score there. So how quick does this happen? It seems like we're talking orders of magnitude speed up compared to the pre-AI way of doing this. And how accurate is it?
Yeah, I mean, RF diffusion, like I can generate a single backbone in a minute, even like a pretty big one. It just takes a minute to generate a backbone. And if you have a really good GPU, that's way faster, right? If I'm just working on my laptop or something, like I can generate something in just like a minute. And then Ligand MPNN is significantly faster than that. So like designing the sequence for the 3D structure is actually really fast. Like per protein and like per sequence, it's less than a couple of minutes to do that whole thing. And so I can design like hundreds of thousands of backbones and then design hundreds of or thousands of sequences for each backbone with Ligand MPNN. So it's pretty fast and pretty computationally efficient. And they've made some improvements to RF diffusion to reduce how many time steps you need to actually get a good structure out of it. And time again, when they actually synthesize these and check for like thermostability or specificity or binding affinity, very often they're very high. And if I have like low thermostability or low binding affinity or low specificity, very often when I use these models to improve that, they improve it dramatically. So they're pretty effective, and they're not perfect, but they're pretty good. Like it's not a hard thing to do to design a protein with RF diffusion and then design a sequence with ligand and PNN and get something that really does fold into that structure with high confidence, or that really does interact with my target protein with high affinity and specificity. Yeah.
So how does this then fit into the broader like validation loop? You have this one thing, you're like, going back to our cancer example, right? This part of the cancer cell interacts with this part of the immune cell and disables it. So we want to put essentially a physical cap on that thing. So that is blocked and that can't happen. And the cancer can get, okay, cool. So I'm going to generate potentially thousands of shapes, thousands of sequences per shape, then potentially run millions of things through an AlphaFold multimer, getting scores. And then what do I do at the end of that? Do I take like the top 100 and go actually try them in a living cell?
Yeah, exactly.
And so this seems, and you're saying they tend to work. And then I guess we also have questions of like side effects would be another big downstream question, right? We don't know what else this thing could do when put into the full environment of the cell.
Well, that's where the specificity comes in, right? Generally speaking, RF diffusion is capable of designing binders, for example, with really high specificity. So they bind to the target and pretty much only bind to the target. Yeah, and it's a really good thing, right? Because if you design a binder that just interacts with a whole bunch of other stuff, it may never make it to the target. It may cause other side effects, like you're saying, have off-target effects. But yeah, RF diffusion is pretty capable of designing really high specificity binders. The sequence or the motif scaffolding, there's more nuance there because you're kind of like performing surgery on proteins and pieces of proteins. And so that can get a little more nuanced and a little more complicated. But if you're just designing a binder with RF diffusion, it's pretty easy to design one that has really high specificity and that doesn't have a lot of off-target effects and stuff.
So what's the bottleneck on this process? Is it identifying targets? Is it especially if you can design things that are that specific and they don't have much in the way of other knock-on effects? I mean, it seems like we should be curing a lot of diseases pretty quick here.
I mean, diseases are pretty complicated, and sometimes it's not just one thing that you're targeting. Sometimes it's multiple targets and multiple kinds of interactions. And so you have to think in terms of entire protein interaction networks, or even interaction networks that involve other molecules as well, and modulating those in very specific ways. And often figuring out what parts of a protein interaction network to change or to modulate and how to do that, what sort of changes you should make to this interaction network. That's a pretty complicated problem. And so I think part of it is identifying specific targets within an interaction network, because some of the interaction networks can get really complicated. And figuring out which parts of them to modulate is not an easy thing to do. And then computationally, there isn't much of a bottleneck. If you just have a good GPU, you don't really need multiple GPUs even. You just have a good H100 or something. You can get a lot done with that, with RF diffusion and ligand and PNN. The hardware requirements are not very taxing. They're probably a little less efficient than I would like them to be, because most average people are not going to have access to an H100. A lot of people are going to be using much lower tier GPUs and stuff. But RF diffusion, for example, there's a Colab notebook that you can run on a T4. You can run it in Google Colab, and it takes a little while. It's not as fast, but the computational bottleneck is not a huge bottleneck right now. I think it could be a little better, but the models aren't huge models. These aren't billions of parameters, right? I think RF diffusion has a couple 100 million parameters or something like that. Or an AlphaFold, same thing. It's a couple 100 million parameters or something. So they're not really big models that require really excessive hardware to do the computations. And I think the slowest part of that pipeline, where you design binders or design or motif scaffold with RF diffusion or partial diffusion or what have you, and then designing the sequence with protein MPNN or ligand MPNN, and then validating with AlphaFold or AlphaFold multimer. The slowest part in that whole thing is actually the AlphaFold multimer part, or the AlphaFold 2 part, where you're validating and predicting the structure of whatever you've generated and designed with the other two models. That's actually the slowest part in the whole thing. If you're able to predict a lot of structures with AlphaFold, then you're fine. There's no real computational bottleneck there. And as far as why are we not curing a bunch of diseases, a lot of this also has to do with how long it takes to get these things to market. And the traditional system that's set up right now for drug discovery and protein therapeutic, it's hard and slow to get new things through. It's a very slow system, and it wasn't designed for large high throughput methods. And it takes a lot of time to get a new drug or a new protein therapeutic through and approved and get it through all the testing, like the different clinical testing phases.
Are we like stuffing the pipeline at this point with new things? I mean, it seems like the biggest reason that this used to be a hard thing, of course, it's like slow and there's a lot of like safety, perhaps redundant steps in there, especially if you could say, hey, we have like very high confidence that this is a highly specific thing. I mean, that would really do a lot to inform what the safety profile might be. But but in the past, it's like the biggest problem was like either it wouldn't work, right? Or it would have side effects that were like intolerable. And if you could take both of those things, not entirely off the table, but if you could sort of say, hey, we can be much more confident now, seemingly like at least an order of magnitude more confident that any given thing is going to do what you expect it to do and that it won't do other things that you don't want it to do. You can improve both of those by an order of magnitude. Then you're sort of seemingly 2 orders of magnitude more likely for things to work, which would take you like from sort of sub 1%, a lot of shooting in the dark, to a lot of the clinical trials would be expected to work. Is that the kind of era that we're entering into now, where clinical trials should go from like roll of the dice to you be more like disappointed when they don't work?
Yeah, I mean, I think we're definitely moving into an era where all this stuff is going to speed up a lot. And I think so. There are like a lot of situations where you want to understand more than just a static structure. Like we've been talking about, like having models that give you more dynamic information and stuff like that. And those are so recent that they haven't really been adopted by a lot of people yet. And I think once those get used more, that'll speed things up a lot and improve things a lot as well. But yeah, I think also part of it is just adoption. Like a lot of researchers are not using this stuff yet because it's so new. It's such a new paradigm. It's such a new methodology. There isn't a ton of information out there about how to use them. A lot of researchers don't understand how to use these models effectively or how to use them at all. Because right now you've got to go through this like somewhat complicated process of setting up these models, and you don't have to know a lot of coding, but you do have to code some, and you have to understand how to set these things up in like a Conda environment or something like that a lot of times. And so if you're not coming from like a programming background, or you don't have some experience with programming, like a lot of these models are kind of hard to touch. They're hard to use because you can't really use them unless you know a little bit of coding at least.
It does seem to be quite a different skill set. I actually studied chemistry as an undergrad.
Yeah.
And the kinds of things that I was taught to do, there were no collab notebooks involved. It was like physical techniques for separating chemicals from one another was a big part of where the time went.
Yeah, and that's a whole other thing too. Like drug synthesis in small molecules is like a whole other story that we can get into. But because there are like really interesting diffusion models out for those as well that'll generate molecules with specific properties. But yeah, it's a very new and a very unique skill set. Most people working on them are still developing their understanding. There aren't very many experts on this stuff. There aren't a lot of people that are training people to do this. I think we're all learning together. I'm definitely still learning a lot of stuff from my coworkers and other researchers in the field. And it's kind of mind-blowing how fast this stuff is developing too. It's developing incredibly fast, which is really good in my opinion. I want to see this stuff proliferated and developed and used because we're going to solve problems much faster this way. There are some platforms that are popping up and are making these things a lot more accessible. One that I would mention is 310 AI is working on a tool that they're calling Copilot, and it's essentially a chat interface that uses tools. So you can talk to it in natural language, like you talk to ChatGPT or something, and then it knows how to use function calling to use other models as tools. So you can say, generate a protein with such and such property, and then it'll use a particular model to do that, generate some protein with that particular property. And then you can say, like, increase binding affinity with such and such protein, and it'll modify your protein for you to increase the binding affinity or something. Or You can say, dock this small molecule to this protein, and it'll call on diffdock and dock the small molecule to the protein for you and then return that. And that's all using like a chat interface, which is making a bunch of these models a lot more accessible to people. And right now, it's pretty good already. It uses a pretty wide range of tools for different things. Like it'll use like protein MPNN or ligand MPNN to design sequences for a structure. I'm not sure if it uses RF diffusion yet, but they just keep adding more tools to it. And they probably are going to start including RF diffusion and some other models as tools for it as well. And once they have a good selection of tools for it to use, that'll be really good. And that'll lower the barrier to entry for a lot of people. But so 310 AI, depending on how good that gets and how fast it gets really good, I think is going to make a lot of these models a lot more accessible and really increase adoption of these techniques. And I think that's going to have a huge impact in the near future. And I think that's going to be really big and important because adoption right now is pretty limited. Just using a really simple metric by, if you just look at like how many views a YouTube video on RF diffusion gets, it's not that many, right? There aren't that many people watching.
This is going to be the one that goes viral and changes it all.
I hope so. That would be great because adoption is going to be big. I think because the more creative people you get using these things, the more you're going to see creative uses of them and like novel approaches to solving problems that people weren't even thinking of before. Like, when have you been able to design proteins that build out complicated protein interaction networks using like binder design and motif scaffolding? Like that's just so brand new within the last couple of years that the adoption just hasn't caught on yet. So hopefully more people will start experimenting with these models and really learn how to use them well. And we'll see a lot of really interesting novel techniques popping up in the near future.
Yeah, it's pretty remarkable. So it seems like the problem then shifts to the case that given a target, we can pretty reliably and like even relatively computationally efficiently come up with something that will hit the target, not hit other things, not cause a lot of collateral damage. Now it's like target identification becomes the thing that really matters the most. And this seems like it's happening in like a lot of areas at once. Certainly thinking about like a military environment, like we've got really good missiles that can hit very precise targets, but then the targeting obviously becomes the high stakes decision. And in lots of just business operations context too, right? Like figuring out the right thing to do is often the hard part. So what do you think are the prospects for that sort of thing? I've been working through this and thinking we've got quite a tech stack for obviously we can like sequence lots of DNA. We can also pull out from an individual cell now what was the sort of state of the transcriptome, like what genes were being expressed at any given time, what proteins were being created. I don't know quite the degree to which we can do that with small molecules with a really localized sample, but it seems like we've got like a lot of ability to generate a lot of data and to probably then create a foundation model of some sort. This is kind of where Evo I sort of sense is going, even though that's not even doing all this stuff yet. But already they're training a language model on DNA sequences, purely bacteria and phage DNA, from what I understand. And
yeah, I was a little disappointed by that, but I think part of their reason for doing such a like specific selection was for safety reasons. I think they're like trying to be a little cautious about what they train on and what the model is capable of doing for safety reasons. But I was a little disappointed that they only trained on that data for sure. Hopefully there'll be another version at some point that's trained on other data as well. But yeah,
I'm sure there's a lot more to come. So they train on all this data. Now can generate sequences in the same way that a language model can generate text, right? In autoregressive byte by byte, base pair by base pair generation. And then there's these really interesting things that they can do downstream of it where, for example, a gene essentiality score or test, basically, if you change a sequence in a particular gene and generate from that changed sequence, it seems that the model has developed a sort of higher order understanding of how things fit together, such that if you do make a change to something that really matters, then you see sort of an unraveling of the later generation. Like you see a very high perplexity downstream of this changed sequence. And that reflects the fact that, hey, if you've changed that sequence, I can't really make any any confident predictions now because we're outside of the set of things that can work. And so once you change that, like it's all kind of noise. Whereas if you change something and predictions remain confident downstream, then you infer that must not have been super important. That was an area that more easily change can be tolerated in that particular sequence because, and we can infer that from the fact that it continues to make confident predictions downstream. That's pretty remarkable stuff that suggests to me that, especially as we scale up the data set a lot more, and this one was 300 billion. I mean, that's not nothing, but it's not much compared to what we could easily imagine doing. Certainly not much compared to the 15 trillion tokens that Meta trained Llama 3 on. But then also like more modalities, right? I mean, we're seeing this in the language models where you can have obviously language integrated with image and plenty of other things. Audio now, it fits into Gemini 2. It seems like you sort of imagine from just the DNA to sort of the DNA and maybe the transcriptome or the proteome or whatever the sort of state of a cell is. And you can even imagine scaling this up another degree to sort of the systems level too. But to learn to predict the next state from the current state seems like we're getting really close to being able to do that. I would kind of expect that in the next couple of years at most, we would start to see large-scale foundation models for biology that would predict like how a cell will evolve through time, how a system level description would evolve through time. And then you could start to do these like counterfactual or hypothetical perturbations and see, okay, if we change this, then how will that make things evolve? If we change this, how will that make things evolve? Right. And so then I guess what we would expect to be learned by those systems is what is now the black box, right? Of all these like interactions that we have only mapped out like whatever, 5% of. And then you could even imagine a situation where you start to do interpretability techniques on digital neural networks to then figure out what the actual pattern of interactions is in the actual biological systems. So is that where this is going over the next couple of years? I mean, and then it seems like if we can achieve that, then you kind of have a fairly closed loop of, okay, we can now identify good targets at a pretty high rate. We can identify or design interventions that are pretty likely to be successful. The like specificity is already high. I mean, we're entering into sort of a super steep part of the S-curve in terms of not just like understanding biological systems, but really being able to intervene in them. And it just seems like a totally different regime that we're headed for. So is that what you basically expect to see over the next couple of years?
I hope so. I hope that all of that comes to reality or comes into existence. I think it's about to get crazy for sure. I think like we're fast approaching a scenario where all of these technologies are going to kind of converge and we're going to have a lot of power over editing and modifying our biology. And I think that's a very exciting thing because there's a lot of problems that I really want to see solved in my lifetime. And I think we are definitely moving in the right direction. And I think we are fast approaching a situation where we can actually solve a lot of these problems. The main thing that you're talking about is like this complicated sort of hierarchical structure that's happening. And the level that I think at most of the time is at the level of like molecule interactions, but you can definitely take that up another level and look at how those interactions come together in a network to create a particular sort of like phenotype or to create a particular sort of state of the organism, right? And like some of those states are diseased states that we don't want, but figuring out like how to modify these interaction networks I think is somewhere that we really need to focus on a lot. And one thing that's really useful and that I really want to see pushed further is this notion of LIS score with AlphaFold and Multimer and seeing Also, like things like AlphaFlow or distributional graph former, generalized to complexes, to like complicated complexes of molecules and not just one or two proteins or something. Like, I really want to see more progress happen there, because once we understand all of the interaction networks and we're able to modulate them in very specific ways, we're going to solve a substantial amount of problems. And like modeling these interaction networks is becoming possible now. I think before AlphaFold multimer and before some of these other like docking methods and before like Rosetta Fold all atom came out, we really didn't have the tools to model all of these different interactions. But we have very recently just hit a point where we do have most of the tools that we need, if not all of them, to model all of these interactions. And once we implement something like AlphaFlow or distributional graph form for complicated complexes of molecules, that's really going to be like pretty substantial. And I'm not sure how I would approach the problem of determining what specific interactions or interaction networks to modulate. Like, that feels like such a big problem to me. But I also know that there are a lot of biologists that know specific interaction networks they want to modulate, and they want to modulate them in very specific ways. And there's a lot of that. And now we can do that. I think we just need to get these tools into the hands of those biologists and make them really accessible, because they're not super accessible yet. I mean, like, a lot of them are open source and they're out there and you can get the model weights, and a lot of them have the training code and the inference code and all this stuff available. But probably your average biologist is not going to know how to use those tools right now. So making them accessible and easy to use is really where a lot of the work is going to be as well, because there are a lot of people that want to do these things, but they haven't quite gotten to the point where they've adopted all of these tools yet. And also figuring out how to use them all in tandem with each other, right? Because you don't want to just use one of these models. You want to use multiple and use them all like together to solve a problem. And so that requires you to learn like multiple different models and how they work and how to use each one of them. And that's not an easy thing to do for a lot of people.
Yeah, this is an issue in AI, even in much more, simple use cases of just using ChatGPT effectively. The sort of the technology diffusion through society is really the big bottleneck, I would say, right now, to when people say, like, why hasn't, if ChatGPT is so great, like, why hasn't it, changed productivity all that much? It's like, people are not using it nearly as much as they could is one really big reason. So
that's a huge reason, yeah.
If I had to kind of map out the story of the next couple of years as I understand it, or as I'm kind of piecing it together from everything that you're teaching me about, it is right now we have a big backlog of targets. And we have a pretty robust new set of tools that used together can design things that will hit those targets and not create too much collateral damage, such that it's not super hard. It's hard to learn to use the tools, but once you have the skill set, it becomes relatively easy to take a target and crunch through a bunch of iterations and come to a bunch of candidates. And those are like likely enough to work that we should start to see serious acceleration of the ability to make these sort of find the solutions to these well-posed problems. And then
for sure,
sort of in parallel, we should probably also expect that Evo 5 or Evo 4 will be capable of dramatically better holistic modeling of the overall networks. And that will then, once we sort of deplete the current set of targets that have been painstakingly worked out through non-AI methods. Why does it always seem like it happens at the same time? It's always these sort of gradual overlapping curves. But my gut says we've got a few years in front of us of picking the low-hanging fruit of, hey, we've got all these targets out there, and now we've got good methods to hit those targets. That's going to take a while for people to learn the tools, do it, obviously validate it, get clinical trials going in lots of different areas. And then as the sort of current backlog gets worked through is probably a better way to phrase it. Right around the same time, I sort of expect that all of a sudden the attention will turn to, oh my God, now we have these foundation models that kind of model the whole causal graph in all of its crazy complexity. And now
Right.
we're actually going one level out and saying, now we can apply similar computational techniques to the identification of the targets. And we'll do that in sort of a similar way of being like, okay, I want this is what's happening and I want to prevent it from happening, or this is what I want to happen proactively that isn't currently happening. Let me just kind of brute force my way through a bunch of perturbations to and of course we can get better than brute force too, but even just imagining a sort of couple generations down of Evo, it seems like make a tweak, see what happens is going to be so radically accessible from a computational perspective that we'll then also just have this explosion of like quality targets to identify. It seems like we may be not super far from we're not even in a world here of like any sort of AGI, right? We're talking like these are still tool simulation and tool type things that people would be using, that we're not assuming anything here about AI agents doing the work. Although you did have a little bit of that with the 310 kind of copilot. But is there anything that I should be like reining in my expectations on? I mean, are there things about like one tweet that I sent you was around to what degree can these things handle sort of point mutations or whatever? And there's maybe individual idiosyncrasy becomes a really hard problem at some point, but like, how far does this paradigm that I'm sketching out extend, do you think, and what limits does it hit?
Yeah, so I think, so just briefly on like point mutations or like more complicated mutations where you mutate multiple things and just predicting how positive or deleterious that mutation or set of mutations might be on the protein or something. Like that's actually that's a capability that ESM has. And that's already 2 or 3 years old at this point. And Evo does it better. They got state-of-the-art performance with Evo on predicting which mutations were positive and which mutations were negative and which sets of mutations were positive or negative. And you can compute a few different kinds of like scores that tell you about this. And you can actually build out evolutionary trajectories to show over time how things are likely to evolve based on how positive or negative the impact of a mutation is on protein or something or DNA sequence or whatever. And Evo, they got state-of-the-art performance on this. And that's actually, they also did this with AlphaFolds. I think they called it alpha missense or something like that. They predicted like all the single point mutations and all the effects that those have. And that's actually like not a hard thing to do. And so like mapping out how a mutation affects like a protein or DNA or something and like the course of evolution that's likely to occur, that's actually like pretty easy to do now. I also want to draw attention to another project that I'm aware of. It's not a model per se, but I think they are using GPT-4. They may have trained their own in-house model. I'm not sure. But there's a company called Future House, and they're designing an autonomous agent that will do a lot of this research for you. And it'll do things like literature search and review and come up with hypotheses of different kinds of interaction networks that you might want to modulate or different targets that you might want. And it'll do it pretty well. That's a pretty new thing that just started happening like last year, I think, maybe late last year. And they're getting pretty good results with that. And that's actually using an LLM like as the base, right, to build an agent to do this research. And then I think they're also building an autonomous lab that drives itself. So once the agent comes up with hypotheses or targets or what have you, the lab is autonomous as well. And that's a pretty exciting direction. I think that coupled with some of these more like specific tools to like perform these like fine-grained operations on like proteins and small molecules and DNA and RNA, when those two kind of converge, that's going to be a really big deal. And I see that happening like in the next year. The progress that they're making at Future House is is really impressive, I think. So that would also be something that people should definitely look into. And like, I think your timeline is within a couple of years for sure. I don't think it's going to be that long before we start seeing like substantial progress and changes. I mean, OpenAI just partnered with Moderna, right?
Yeah, hundreds of GPTs.
And I think
I'm sure
that's
just the beginning.
And I think that also is like a paradigm that's going to match well with these other more specific tools, because when you enable an agent to use tools, you unlock a lot of possibilities, right? When you enable something that can review thousands of research articles, then develop like targets or hypotheses to test, and then can call on these specific tools to design molecules or proteins or nucleic acids to perform these specific functions or hit these specific targets, that's going to move really fast. And I think I'm excited. I'm also a little nervous because I think there's a lot of potential for misuse in the wrong hands. Like, the sort of things that you can accomplish will be amazing and beautiful, and we're going to see a lot of health problems just disappearing. We're going to see lifespan extended, and that's going to really improve the quality of life for everyone. But also, we do have bad actors in the world. And that is something I worry about for sure, because in the wrong hands, we could be looking at very dangerous things as well. And so having some kind of oversight for these things is very important, because, I mean, we're looking at moving into an age where you could target a specific group of people based on their genetics or something. That's both immensely useful and also very dangerous. And I have a lot of faith in the people that are working on these things. Like, the people that are building these models and doing this research are really good people with a lot of really good intentions and a lot of know-how and experience. And that to me is very reassuring. But there's always some random jerk that has the potential to mess it up for everyone. We have to be prepared for that.
Yeah, no doubt. I mean, the preparing for this, definitely one of the major updates that I've made is that when people talk about the bio risks from AI, the conversation that I've heard most of has been like, how does it compare to Google? How does GPT-4 compare to Google? Does it make it easier for you to get certain information or figure out how to do certain things? And in familiarizing myself to the degree that I have with all this technology, it's like that all of a sudden feels very quaint already. It's like this is not a question of comparing to Google. This is like generating entirely new stuff. And I looked back not long ago at the list of mass extinctions in the history of the planet and what caused them. Of course, some were caused by totally exogenous shocks, like asteroid hits the Earth. But the first one on the list on Wikipedia is the oxygenation of the atmosphere. And it's simply that something kind of pops up and either itself, nobody knows how to eat, or it creates some waste product that, that nobody's prepared to deal with. And what we now breathe and depend on was at one point the cause of a mass extinction event. I kind of try to keep these like super zoomed out perspectives in mind. And it does seem, I mean, tell me if you think there's any limitation to this or whatever, but with this sort of brute force search through sort of biological space, right? It seems like there's not really anything conceptual that I could identify preventing, talk about gain of function type research on a totally different level. Those seem like the really dangerous things. And I don't know how you prepare yourself for that.
I think there are multiple things that we can do to help prevent some of these bad things from happening. And I think we're going to need to rely on the models that we build, especially the agentic models that we build, to help guide us in some of this. Because at some point in the near future, we're going to have agents based on really robust language models or something similar. And they're going to be able to review thousands of research papers and do tests in a lab on things, take in an amount of data that we can't really take in, right? That no human or even group of humans can really take in and process and digest and use. Like the scale is going to be much larger than a human can really work with. And so we're going to have to rely on some of our models to help guide us. And also, like going back to how does it compare to Google, I think another argument that could be made for why not to worry about some of these things is, okay, maybe I can get on my computer and design some new thing that was really toxic or something, but I still have to go into a lab and synthesize all that stuff. And a lot of that part of the process is very highly regulated and watched, right? I can't just go get a bunch of random chemicals and build this stuff in my house, right? Like it's harder to do than that. And like synthesizing proteins it's a non-trivial process. So like a lot of the worry, I think, of, oh God, we're going to have an AI that designs a deadly virus or a bioweapon or something. A lot of that is really overblown, especially at the moment. And I think a lot of people overlook the fact that computationally designing a molecule is just one step in the process. You also have to do all the lab work and synthesis and work on some kind of delivery mechanism. And this is all stuff that's non-trivial to do. And that helps prevent bad actors from actually going through with some of this stuff. Now, there may be like state-sponsored bad actors with access to good labs and lab equipment that will circumvent a lot of that. But when you're working at like a state-sponsored level, like that's a matter of like international relations and also national security. That has almost nothing to do with AI, right? Like the computational design of some random like toxic molecule is just one step in a complicated process. And people don't often think of that. And I see a lot of like very influential, big name people in the industry who are coming from like the NLP side of things and like the LLM side of things. And they don't really understand the biology and the process that goes into actually making these things. And I think their worry is justified, but they're also like not understanding the nuances and where the dangers actually are. So that I think it's important for people to keep that in mind. I think it's very important before we start getting all anxious and worried about some random person using Llama 3 to design a superflu or something. You have to remember it's very unlikely and very difficult for just a random average person to go through the entire process of designing and synthesizing and delivering some kind of toxic molecule. That's a long, difficult process that an individual would be very unlikely to be able to accomplish, even with the help of a very intelligent LLM or a very capable agent. Now, when you have larger research-oriented companies working on these things that have like their own wet labs, and they're building agents to run these wet labs and to computationally design the molecules and form plans on how to use them. Like there, I can see, okay, we need some kind of oversight. We need some people working on how to make sure that process is safe, how to protect that information and data and equipment, right? Because there are plenty of situations where something like corporate espionage is happening and people are trying to do nefarious things with some of the technology that some of these companies have. But again, that has very little to do with the AI itself and has more to do with how we're interacting with other countries and other research organizations. So definitely we need to be developing plans for how to use and regulate and oversee like a really capable agent that is going to go through the entire process of like computationally designing and verifying and then also synthesizing and delivering. And that is technology that is like in existence now. We already have agents that are like doing most of this process, right? And a lot of people are pushing for more of that, which I agree with, because we're not going to be able to solve these problems on our own. We're going to need some kind of really capable agent that can help guide us through these processes because they're so complicated and so difficult to understand the whole picture and all of its nuances that I don't know if humans can get there without some kind of agent helping out and helping design the molecules and producing it and delivering it, and et cetera. My concern is just making sure that these companies and organizations that are building this technology and using it have some people that are overseeing the safety side of things and that are concerned with red teaming and preventing things like corporate espionage and making sure that everyone that's using that technology is using it responsibly, and making sure that that the companies are hiring not just capable people, but also like people with good moral grounding and good intentions. That's not even the future. That's kind of now. That's already a concern that we need to address now, because we already have agents that are doing these things. And most people don't have access to them, though. Like, most people don't have access to a really capable agent that can do this whole process or do most of this process. Like, that's not something that even most companies have access to, much less individuals, because a lot of these models are closed. And I could see like maybe some kind of like state-sponsored group of researchers could use open source models to build something similar and do something nefarious, but that's a whole like, it's a very complicated process of building such a thing.
Do you think that do we have any read on whether offense or defense is favored here, so to speak? In the sense of, for nuclear weapons, for example, if one of the major nuclear powers fires all its missiles, nobody can shoot all those missiles down. It seems to me that is an offense-favored regime, and so we're kind of stuck in this like mutually assured destruction paradigm, which is yikes. We kind of need to get to a different paradigm because we're under like real threat of nuclear catastrophe as long as we've all got thousands of missiles pointed at each other and no viable defense. I don't have an intuition for I think if biology like works the same way or not.
I think this is like less of a question about biology and more of a question about cybersecurity, because I think it's like very similar in spirit to like cybersecurity. And there as well, most often the attacker has the advantage over the defender. But there are a couple of things that may change that. One is really capable agents. Because if you have a really capable agent that's able to defend against human attacks that are like just inferior, that's going to be a really big part of protecting against bad actors. And so I guess like that's an argument for acceleration, actually, because if you have the most capable agent, then probably your defense is going to be a lot better than everyone else's. And if your agent is capable enough, it may be effective enough to ward off pretty much anything. And I don't know how soon that's going to come into the picture, but I think that is a good argument for keeping up the pace of development of LLMs and agents and things like that. I think the advantage of the attacker over the defender may end up shifting because of agents. We may end up having a situation where that's no longer the case. And
so I'm a little confused about why is the shift from biology to cybersecurity, because I'm envisioning a world where, for example, Evo is open source, Llama 3 is open source. If Evo 3 is open source, then we start to, at some point, we enter into a regime where, it may not be easy, it may be, hard for one person, but at some point it does get like lower than state actor level where somebody could launch some crazy attack. And then it's okay, if you create some superbug with certain properties, can we defend against it? I'm a little bit unclear as to how I'm not sure if you're like equating that to cybersecurity or saying that's like primary somehow.
No, I think that's a good point. Yeah, because if you develop some kind of, I guess a virus is probably as good an example as any, but let's say you develop a virus of some kind that targets a specific population. Developing a cure for that traditionally has been a very slow process. And like the fastest that we've ever done it was probably COVID. And that still took some time, right? And on the other hand, there is recent work that came out of, it was the University of California, but I'm not sure which one. They recently published some research about like universal vaccines. and they're able to design a vaccine that was like applicable to a wide range of mutants of a virus. And they said that the technology or the method was highly transferable to other vaccines. And so there's no reason that this can't be applied to pretty much any vaccine to develop like universal vaccines against all variants of a virus, or like most variants anyway. So that'll be helpful. That'll be good for like defense sort of things. And then as far as the applications of certain AI models to developing defenses, it's a very complex topic that some of the new things that are coming out feel like they might be the answer, but it's a little too early to tell.
Yeah, that's definitely that's a very good data point. I mean, my kind of default would be just to think, I have 3 kids and I'm no expert in how babies develop in the womb, but it's definitely clear that a lot of things have to go right. Like an unbelievable number of things have to go right in the proper sequence. At any point, if something goes wrong, like that could be the end of it.
Yeah.
My general default model would be like a lot of things have to go right and only kind of 1 or 2 big things would have to go wrong. And so it seems like there's a lot of surface area to defend and a lot of kind of places that could be attacked. But then, hey, if you can make a universal vaccine, then all of a sudden that does start to look quite a bit different. And I think you're right also to say a big part of this does seem to be sort of what is the prevailing like international relations regime, because if it seems like pretty safe to say, if we get into a bioweapons arms race, we're going to be in bad shape. We really have to have some more globally cooperative approach. Or the missiles have one really nice property, which is they don't like spontaneously escape their silos and self-replicate around the world. Whereas the list of lab leaks is like quite long.
Yeah.
It just seems like there's no way that we can get into an international bioweapons arms race and survive it. We just have to avoid that trap in the first place. And
also,
I hope we do.
What happens when you develop cures for a wide range of diseases and you're able to extend human lifespan significantly longer than what it is now and eliminate a lot of the diseases that we face? Once that exists, I don't know, maybe that's a paradigm shift. Maybe that's like a shift in human consciousness at that point, and we start thinking about things very differently, because we're all used to thinking about like everything in terms of being finite. And I think having an approach or thinking about things in a way that isn't finite anymore, and thinking about things in terms of how valuable our health and our life is, because of the fact that enables us to be with who we love for longer. The time that we have with the people we care about is, in my opinion, the most valuable thing that we have. And once you enable people to have healthy lives with people they care about, more or less indefinitely, that changes a lot. And I'm very excited to see. I hope that happens in my lifetime. I think it will. I think it'll probably happen within the next decade even. But I hope that really changes human consciousness to a point to where a lot of these problems like just kind of start to go away, because we stop thinking of everything in terms of finite resources, finite lifespans, finite time with the people that we care about. Like, hopefully it's enough of a conscious shift in consciousness that we see some of these problems fading, because a lot of them are cultural, right? a lot of these problems, or a lot of these like threats at the heart are very cultural. It's not about the technology, it's about how we use the technology and how we're interacting with each other when we use it. And that requires people to think differently. It's not a problem that can just always be addressed with some new technology or some new defense mechanism. We really have to change our thinking. And that, I hope that when these things start becoming widely available, people's thinking will shift dramatically. Maybe that's where things are headed. I actually have a lot of hope that's where things are headed, and a lot of optimism, because I think overall, like, most people are good. And I think like we can heal a lot of things, not just health problems, but a lot of psychological things we can heal with these tools, because it's going to change the way that we interact with each other and the way that we perceive our environment and our relationship to it.
That is a beautiful sentiment and maybe a good place to end.
Yeah, I think I agree with you. This has been really great, by the way. I had a lot of fun doing this, and I really appreciated this.
Well, I appreciate you teaching somebody who doesn't know nearly as much as I suddenly feel like I really should about this area. So feelings definitely mutual. Amelie Schreiber, thank you for being part of the cognitive revolution. It is both energizing and enlightening to hear why people listen and learn what they value about the show. So please, don't hesitate to reach out via email at tcr@turpentine.co, or you can DM me on the social media platform of your choice.